video recorded. Anyway, so classification and regression trees is the title of a book that was published in 1984 by Bryman, Friedman, Ocean, and Stone, two Stanford and two Berkeley professors. But the concept goes back all the way to 1954. So we are more than 50 years, more than 50 years since the original concept of a classification tree came out. And I started work on this in 1985, which means I've had 32 years of time thinking about this topic. And I wrote a paper by the same title, Classification and Regression Trees. So if you Google this term, classification and regression trees, which I just did, you'll see that you see the Bryman book, which has 32,000 citations. Then the next thing is you get to see my paper, which has 481 citations, which is this article. So it's a top ranked by Google on this subject. It's just an introductory article. It's like some sort of encyclopedia article, about 10 pages. So if you're wondering whether you want to come back after today's lecture, you can read this article and you may find that, gee, this is so easy, I won't come back anymore. Or you may want to think, you might see that, oh, actually there's a lot more to it. So this paper of mine I published in a journal called Wire's, Wiley Interdisciplinary Research Series. And people, when people Google this, they will read my paper. And on average, 10,000 people read this paper a month. I don't know whether they really read it. Maybe they download it and trash it. But 10,000 hits a month. So this tells you how popular, at least people think this topic is popular. And I have my own algorithm. So if you go to my website, which is the Wisconsin website, so if you go to like the first page, so you go to www.stat.wis.edu/guide. Don't type in the IMS part. Okay, you type that. Well, if you just type ~low, if you just type ~low and end there, you get this picture of me when I was very young. It was professionally taken, and I never had a professional photographer again, so I'm reluctant to replace it. So when people Google this, people think this is a teenager. But you go to research, okay, and then you see guide, right? So I'll be talking mostly about the algorithm called guide. It is now in version 26. I started writing it. I released it about 25 years ago. So about every year or so, I release a full version. In the meantime, I release fractional versions. And if you want to know whether people use these kind of things that I do, on this link, it says application papers on Cruise, Guide, Fact, Lotus, Quest. These are all algorithms that I developed with some students. But Guide is completely developed by myself. I develop it and I write the code. 20,000 lines of code. I'm pretty proud of. Took 25 years to write that. So if you click on that, you see a list of papers that people have published using the algorithm that I have written. And these are people who never heard me say a word. I've never met them. They go to my website, they download the software, they download the manual, they understand how to use the software, and they publish papers. So the first paper is published in Mathematical Methods in Medicine, and then another paper, second paper is published in Pediatric Blood and Cancer. I guess it's easier if I read here. Pediatric Blood and Cancer. Another one here is very simply gut. Gut, I guess, means stomach. And then BMJ Open, I don't know what that journal is, maybe medical journal. Analytical Letters, Global Environmental Change, Proceedings of the National Academy of Science, Remote Sensing, Procedure, Environmental Sciences, Emerging Technologies, Sustainability. So, there's almost 200 papers. I stopped updating a few years ago. But so just to let you know, okay, what you are here for. In this tutorial for this week and next week, you'll be hearing from me about things that I've done. And you cannot hear the same thing from any other person. Because only I can tell you the story. All right? And so that's some background about this work. So that's the application papers. But you click on guide, that's where you can get the software. So 3 years ago, I did a tutorial here, and we went to Sentosa on the weekend. And you recognize Sentosa? I don't know what this is, Sentosa. So some of my former students who were at this workshop took a picture. So this page is about guide, tells you some of the properties of the algorithm. And then it tells you the documentation behind it. So this is in reverse chronological order. The first paper published last year in Statistics in Medicine. Another paper a year before that, that's Precision Medicine. We were talking about that, not today, but certainly before halfway through. And then I wrote a more serious review article of the subject, 50 years of classification and regression trees. So if you go to here, you can just click on it, you can download the paper, you don't have to go to the journal. So these are all these papers written. My first paper was in 1988, JASA. If you are curious, you can read that. It's a discussion paper in JASA, and I got trashed by Bryman and Friedman for proposing a shortcut algorithm. Okay, but well, 25 years or so later, we're here, 30 years later. So now these are the compiled binaries. So the software is free. The source code is not free. I get all kinds of requests all the time asking if I can have the source code. People are saying, I'm writing my PhD thesis. You have a wonderful method. It would be lovely if I can have a source code to add something to it for my PhD thesis. And my attitude is, to write a PhD thesis, you write the software. You don't take somebody else's software, modify a bit of it, and make it your PhD thesis. So these are executable binaries. It means they're exe files, right? You can use it to run. It's free. And in fact, drug companies are using this nowadays more and more because, well, later on when we talk about precision medicine, I'll tell you more about this. But in precision medicine, we're talking about a lot of variables, a lot of biomarkers, and we want to try to understand the kind of models you fit, and tree-structured models are the easiest for people to understand. So one drug company, one major drug company, for example, that I work with, originally I would email them every time I have a new version, and a new version comes out quite often, maybe every few weeks, maybe every few months. They're sort of decimal versions. And after a while, they decided to create a web crawler. So they have a web crawler, which is an automatic piece of software that every night would check my website for the latest version and download it immediately, and they use it company-wide. Okay, so I mean, this is serious stuff. This is industrial strength. This is not toy. It's kind of software. Okay, I mean, serious companies are using this. Of course, I don't get paid a lot for them to use it. Any kind of payment is fine. This is free. Right? By the way, you're not paying me to come here. Some of you are paying, but you're paying IMS. And those of you who are paying, I ask them, can we have better cookies? They say, so anyway, but we do have better cookies, apparently, because some of you are paying. The rest of you just enjoy the cookies. So, all right, so I have Linux executables. I have 3 Linux executables, one compiled with Red Hat, no, they're all, Linux is Red Hat and Ubuntu, so I have one compiled in Intel Fortran, one compiled in NEG Fortran, one in G Fortran, and if you're a Mac person, there are 3 versions of Macintosh version compiled with AppSoft Pro. Oh, by the way, this is written in Fortran, but it's not your grandfather's Fortran. Okay, this is C++ type Fortran. This is Fortran 2015. Okay, when I was a student like you guys, I was using Fortran 77. But this is Fortran. This is really top-notch Fortran. So that's Macintosh. And for Windows, I have 4 versions, 364 bits and 132-bit, right? So you may wonder, why do I have so many versions? Because I'm really serious about software as well as I'm serious about concepts and theory. If you are really a computer person, you will know that when you use a compiler, right, some compilers are more rigid than other compilers. So for example, if you have a vector that you think has 4 elements, right, but in your software you have made a mistake, and it tries to retrieve the 5th element of that vector. Okay, so this vector in the computer has only 4 elements, but somehow you make a mistake and it's trying to access a 5th element. Many compilers, including G-Fortran, will not trigger an error, will fill some garbage. And how many of you use R? Many of you use R. That should scare you, because R is compiled with G-Fortran. Okay, but maybe that's okay, because there's really so many bugs within R itself that G-Fortran. So I make a lot of versions on even the same platform, so that if there's a bug, right, one of these compilers is going to choke, and it's going to crash, and I know there's a bug. Okay, so you have a choice about this. But if you're a Windows person, how many of you are Windows persons? I see a Mac person here. How many of you? Oh, only 4. How many of you are Macintosh people? Okay. The rest of you are Linux. How many of you use Linux? Okay. Does that include everybody then? Is there anybody who didn't show his hand? Anybody else use it other than these 3? Okay. So, okay. I thought most people use Windows, because in this industry, everybody uses Windows, at least in the drug companies. So I brought my Windows laptop in case. So what I'm going to do is I'm going to interlace these lectures with a live demo. Okay. So in the past, like 3 years ago, I gave something like the same thing 2 weeks. And I went on and on and on with the slides until the last day. Then I spent 1 hour, half an hour, right? But that time is too late. If you're really interested to use it, it's too late for you to try it, and you run into trouble, you ask me, because I'll be gone, right? So this time I decided that I'll start to demo a little bit of the software the next tutorial. I'll give you time to download the software, right? So you go to here to download it, right? It's just one file. There's no installation. You just unzip it. And there is a manual. So this one, right? There's a manual. So if I click on it, that's the user guide. And it will tell you everything about installation and so forth. Right? So if you're serious about playing with this software, download it, download the manual. So on Wednesday, I'll at least walk you through some basic examples, right? So I think it's more enjoyable than if you just listen to me talk. Just underneath this link, guide manual, there are manual example files. So the manual has a lot of examples using real data sets, and those data sets are in this zip file called datafiles.zip, right? So if you are really interested, you want to follow with the demo next time, you download the manual, you install the program, and then you download this, and you have all the data files that I will use for the demo. So you can actually, go along in real time with me. All right? Okay. So I have some older programs which I will not talk about. All right. So that's the background. So my second slide tells you something about guide, which I already told you quite a bit already. So there's some papers. There's my first paper, and then there are a lot more papers, but specifically these are the key papers. And this research is supported by both government and industry. All right. So here's the outline of the tutorial. We have 5 days of tutorials. So I will do it sort of like tutorial by example. There's a book called Regression Analysis by Example, where you take a data set and then you illustrate some techniques in regression. So I will follow the same style. I'll take a data set, a real data set, and I'll tell you how difficult it is to use traditional methods. Okay, and for some of these examples, you just can't do anything else. And then I show you how you do it using guide, right? So the first example is glaucoma, predicting whether a patient has glaucoma based on some images you take of the eye. It's a very old data set. And we're going to build a classification tree. So a classification tree is a model that predicts whether somebody has glaucoma or not, right? So you go for an eye check, the doctor looks at a lot of things in your eye, and based on that, he or she will say, I think you're okay or normal. Otherwise, the doctor, I think you have glaucoma, right? So that requires a doctor. What this does, some people call this machine learning. You have heard of machine learning, right? It's a computer program that takes a data set of patients with glaucoma as well as without glaucoma, and for each patient, they record a lot of things about their eyes, all right? And try to learn from the data set to come up with an algorithm. They will predict for another patient, right, whether the person has glaucoma or not. Of course, it's not going to be perfect prediction, but it usually is pretty good. The second one is, so the first example is you're predicting glaucoma status based on continuous predictor variables, explanatory variables. So those variables are continuous. In the second example is predicting whether a protein or something binds to a certain molecule. So in drug discovery, you may find that a certain disease produces certain kinds of proteins or something that will be attracted by a certain molecule, right? And so if you can attach this molecule as an attractor to something in your body, then you may be able to kill this bad stuff because it tries to attack this, tries to eat this, and this thing is poisonous, something like that. So it's called peptide binding. And this is an example where the predictor variables are categorical variables, like DNA type categorical variables. But this is very high-dimensional categorical levels. Then I will use the heart disease data to show some differences between CART and GUIDE. CART stands for Classification and Regression Trees. It is an abbreviation of that book title I mentioned to you that was published in 1984. It is also the software that came with that book, right? CART is still being sold for a lot of money, but CART has never been revised since 1984, okay? So CART is like 1G and we're like 5G, okay? Except that 1G is very expensive, 5G is free, okay? But nonetheless, if you want to know about the history, you have to know about GUIDE, you have to know about CART. And I'll use an example on heart disease to demonstrate why CART is 1G. And we all know 1G is slower than 2G, 2G is slower than 3G, right? And 1G is, as far as CART is concerned, is really, really slow. And then the third item will be Alzheimer's disease. Okay? Alzheimer's disease is, I think, the most attractive disease that drug companies are targeting right now because it's guaranteed to be very profitable because there's no cure for Alzheimer's, right? So you can only hope to slow down the Alzheimer's. Alzheimer's is a deterioration of brain cells, right? You cannot cure it except to do a brain transplant. But you don't want to do that because if you do that, you're a different person. It's somebody else's brain. Why do you want to put somebody else's brain in your body, right? So Alzheimer's disease is a very profitable disease, but it's very difficult. The medical community does not yet know what causes Alzheimer's, right? They can see some correlations between something called plaque in the brain. They call it tau. It's some plaque in the brain. They find that Alzheimer's patients have this kind of plaque. And so because they don't understand what's causing Alzheimer's, because they see the correlation, they now find drugs to attack the plaque. Okay? They hope that if the plaque is the cause of the disease, then if you reduce the plaque or get rid of the plaque, then you don't have the disease, right? But apparently this theory is not really that good. So a few months ago, it was a big news in the United States. Two drug companies, Eli Lilly, I think, and Merck, right, had two very promising trials to demonstrate the usefulness of their drug, and both collapsed. Okay, both trials turned out to be unsuccessful, and they lost a lot of money, lost a lot of time. So I'm going to use that to as an example for longitudinal response. So In the first few examples, we are trying to predict whether you have glaucoma or you don't have glaucoma, right? But in Alzheimer's disease, the response, the dependent variable, is time dependent. You observe the patient over time. It's not like glaucoma is different. No. Do you have glaucoma now or not, right? But in many of some of these diseases, people watch, like prostate cancer, right, or even some other types of cancer. The patient comes in every few months for a checkup, and they see whether the cancer is grown or not, right? How bad is the Alzheimer's disease? And so the Y variable is a vector, right? Y1 is your value, your stage of your Alzheimer's disease, or seriousness of the disease at the first day of the trial. Y2 may be 6 months later, Y3 may be another 6 months later, right? And it goes on for maybe 5, 10 years. So now your response is a curve, okay? And you will see that some people with Alzheimer's, the curve goes up very steep after some time, right? The higher it is, the worse the disease. And people without Alzheimer's disease are sort of flat, right? So the idea is, can you find a drug that will slow down the rate of increase of this curve, right? So now we have longitudinal response, not just one Y value. And then after we're done with that, so items 1 to 4 has no treatment variable. In precision medicine, you're talking about a drug, and when you do a clinical trial, you compare the patients given the treatment and the placebo, patients not given the placebo. In items 1 through 4, we don't talk about the treatment yet. Everybody has no treatment. We just watch what happens when you grow old and your Alzheimer's disease gets worse and worse, right? But from item 5 onwards, we talk about subgroup identification. Subgroup identification is to find a subgroup of patients for which a drug will benefit them much more than average, right? So one of the examples I'll talk about is breast cancer. Okay, it's quite established by now that any woman post-surgery after breast cancer is given a hormone therapy because many breast cancer cells eat estrogen to grow, right? So the purpose of the hormone therapy is to have the patient eat a fake estrogen, okay? So the breast cancer cells think they're eating a real thing, okay? So this is like an alligator or a crocodile, right? If the alligator or crocodile is a rock, right? And if the rock is big enough, then the rock cannot go into the stomach, but cannot come out. It's stuck in the mouth. If that happens, the crocodile will die because it cannot eat anything. It will die. So the idea of hormone therapy, one of the treatments, is this fake estrogen that makes the breast cancer cell eat it. Once the cancer cell eats this fake estrogen, it cannot eat anymore, and it cannot throw it out. And then the cancer cell will die because it doesn't have the food, right? But not all breast cancer cells are like that. Quite a number of breast cancer cells are like that, but some breast cancer cells do not require estrogen to grow. They grow because they eat something else. Subgroup identification is to identify the type of the women who have this kind of cancer. So you give this kind of treatment, all right? The women with other types of cancer that don't eat estrogen, you should give them some other treatment, all right? But right now, right now, most of the time, every woman is given hormone therapy, regardless, right? And that is found that on average, when you average across the patient population, right, the benefit is good enough, right, for the Federal Drug Administration and so forth to approve it. But then there are some women actually who don't benefit from that treatment, okay? And so if you can exclude this group, so if the average benefit is X, and suppose half of them do not benefit from this treatment, if you remove it, then the benefit for the remainder will be 2 times X, right? If the average is X, and one group has 0, the other group must have 2X to average to X, right? So this is what's going on in the United States in drug discovery, because nowadays the diseases are very difficult to treat. For example, cancer, because cancer evolves, right? You try to kill it with something, you don't kill it, it will become immune to this drug, and you have to have another drug. So as a result, if you look at average benefit, it's very hard to get a drug approved by the FDA. They will say the average benefit is not large enough, okay, for the drug to be approved. So the drug companies now they all use this different strategy, is to find a subgroup where For the average for this subgroup now is 2 times X. And once they can find a subgroup, they do a clinical trial and that prove that the benefit is 2 times X. And they get government approval, right? And when they get government approval, on the label of the drug, it will say, well, this is only for people with this HER2 gene, right? For breast cancer, there's something called HER2 gene. You may have heard of, but have you heard of Angelina Jolie, right? Angelina Jolie, the actress, she has that, and she removed both breasts, okay? Because that gene is terrible, right? So there's some drugs that say on the label, this is only for this, or only for this kind of gene mutation, okay? And you go to that drug on the website, and you go to the drug page, you can download the PDF file on the label, and it says this is a group of women that you should prescribe, right? So how do you find out what group of women would be on this label? That's subgroup identification, okay? Of course, don't get too excited. I'm not going to show you some wonderful drugs here because I don't have kind of data. Even though I collaborate with industry, I don't have the kind of data about. So we'll just be talking about things that are public domain. So breast cancer is one example. And then this is another example where you have genetic variables. Okay. Now, one of the things which is unique for this algorithm guide that I'm talking about to you is that it can handle missing values, right? Very few methods can handle missing values. How many of you have heard of lasso? Okay. Lasso is like a linear regression model when you have a lot of variables, right? Everybody thinks lasso is great, right? And a lot of theorems are proven about lasso. Lasso cannot handle missing values. If you have missing values, all the theorems will not apply, right? So logistic regression is great, isn't it? How many of you have used logistic regression? Right. What happens when you have missing values? You throw away the observations with missing values, right? Well, in this example, this is not logistic regression, but you throw that away, you have nothing left. Okay? It's not that every subject has a lot of missing values. Every subject only has very few missing values, but there are a lot of X variables. All you need is one X value to be missing, and that subject will have some missing value, right? So missing value actually is the most difficult problem in statistics. You think that subgroup identification is difficult. Yes, it's difficult, but it's solvable. Missing value is unsolvable. Given a data set with missing values, there's no way you can tell why the values are missing. There's no way. The data doesn't tell you why certain numbers are missing. So all these missing value methods, missing value imputation, requires a model, right? You model the missingness, and then you do this imputation, okay? If your model is correct, okay, great. Then you can apply other things after that. You can apply lasso, you can apply even, have you heard of random forest? Many of you have heard of that. How many of you have used random forest? Right? Yeah, you like it, right? Random forest cannot handle missing data. All right? It cannot handle, our package does not. Our package has a function there called NA fix or something like that. You call that, it fills in all missing values with medians. And then you go ahead and do a random forest. If that first step is wrong, there's no guarantee that subsequent steps will be good. You really screw up your data, right? You really mess up your data. Okay? One thing with guide is we don't need to do missing value imputation. Okay, so you save that first step, the most difficult step. Then we apply this to longitudinal response for type 2 diabetes, right? So this longitudinal response, Alzheimer's disease is longitudinal response, but here we have a treatment, okay? Drug A versus drug B. For Alzheimer's, we don't have a treatment. Nobody actually knows about any treatment. In fact, I've been consulting with some drug companies, and they're trying to design a clinical trial, right, for a drug for which they do not know whether it works or not, but they have to do a clinical trial because they say that in the lab, it worked. So what did they do is they took a monkey and damaged this monkey's brain, and they give the monkey this drug. And then the brain grew back. I asked, how many monkeys do you have? 1. Okay? You have a sample of size 1 where you demonstrated that this drug worked. Okay? And now they want to do it on human trials. Okay? They don't have any idea whether it works on human beings or not. But it's a race. All the drug companies are in this race, right? Whoever comes out first will get billions and billions of dollars, right? So they asked me to help them design a clinical trial for this drug for which they don't know what is the effect. So they have to first do simulations, right? So you simulate the Alzheimer's trajectory for ordinary patients without the drug, and you simulate the trajectories for people who are treated, right? It's very difficult when you don't even know what is the treatment effect, right? So you try, or suppose the treatment effect is to bring down the slow down by 10%. This is the sample size you need, right? Suppose the treatment will bring down the slow down the thing by 20%. This is the sample size you need, okay? So so anyway, we're not talking about Alzheimer's here. We're talking about type 2 diabetes, because now for this one there are a lot of diabetes drugs, and we can discuss that. And then the final thing, okay, which I'm quite proud of, because I have been unable to solve it for 30 years. Okay. So you have so for those of you who know about lasso, right? In lasso, it's a linear regression model, and you have more x's than you can fit, right? So the lasso model tries to make some of the beta coefficients for those some of the x's 0, right? So in the end, you only get a subset, right? So maybe x5 has a non-zero coefficient, x10 has a non-zero coefficient. And you can estimate those coefficients, right? But what people want to know is, what is the standard error? What is the variance of that beta 1 hat if x1 is in the model? What is the standard error of beta 10 hat, the standard error? You cannot use least squares, right? Because there's a selection phase before that. This is called post-selection inference, okay? So there's a lot of people I know, they're trying to solve this problem, especially mathematical statisticians. These are lovely problems to solve because it's a linear model, all right? And they try all kinds of methods. In my case, we have a tree-structured model. And so we'll find a subgroup, right? So we say a subgroup will be, say, people with this genetic marker, a certain age, a certain race, some history of this disease. And then you find that the benefit is 2 times x, okay? The doctors want to know a confidence interval for 2 times x. For 30 years, I thought it's impossible, because this subgroup you find, it cannot be replicated. You take another set of data from the same population, you apply the algorithm, you will not get the same tree. So that subgroup will change, right? So it's more difficult, because now I have a non-parametric model. In the lasso, you have a parametric model. I have a non-parametric model, right? But people want to know, okay? You say that, okay, with this treatment, your rate of disease is slowed by 50%. What is the standard error? Is the standard error 25% or 30%, right? Or is it 5%? So people want an estimate of accuracy. So I'm happy to tell you that last year I solved this using a bootstrap, okay? And that method that I used to solve it, I actually published a paper in 1987. So 30 years ago, I published a paper in JASA on bootstrap confidence intervals. Nothing to do with trees, nothing to do with precision medicine, okay? And then after that, I worked on this thing. And so didn't think about applying that, okay? So that's item number 6. Item 7 is something that was told to me by some very smart statistician in a drug company. So after I developed this method to find a subgroup of patients, and so suppose you find a subgroup, these are the people with this biomarker levels and this gene and so forth, right? And then I find that, oh, the response is 2 times x. Well, this very smart statistician asked me this question. He said, in that subgroup, is that confounding with other variables? Right? Now, before you do the subgroup, you take the whole sample. There's no confounder because it's a randomized trial. Right? In a randomized trial, all the covariates are balanced by chance. But now you do a subgroup, right? You mine the data and get a subgroup. Might that balance be destroyed? So in other words, if the balance is destroyed, you see this benefit. Maybe this benefit is because in this subgroup, everybody turns out to be young, right? So we know young people survive longer than old people, right? So maybe most of these people in this subgroup who are given this treatment that looks so promising turns out to be young. So it may not be just a drug. It also could be age. That's confounding. Right? So how do you control for that? Okay. So we'll talk about how to do that in item number 7. And then I'll compare with some other methods that have been proposed earlier. All right? But none of these methods can do what we can do. Okay, so let's start with our first example, glaucoma. So this is a data set in this package by Thorsten Hawthorne. Some of you may know this guy. He writes a lot of our packages. So that package is TH, is his initials. That package is our package. It doesn't have any routines except data sets. And actually, this data set is not from them. This data set is already around for a long time. He just put them together, right? But if you want, you can get it from that package, or you can go to my website and download the data files.zip, and zip it is there, right? So there are 170 patients, 17 with some missing values. And there's a Y variable, right? So the Y variable is whether you have glaucoma or you're normal. So the Y variable is like 0, 1, or you just call them glaucoma or normal, right? And there are 64 predictor variables, right? What they do is they do something called confocal laser scanning of the optic nerve head, and they do something also called fundus photography. I really don't know what these are, but just think that we have 64 measurements of the eye, okay? We have 64 measurements of the eye for every one of these 170 people, and some of them have glaucoma and some of them are normal, right? So obviously 64 X, think of them as X variables, right? You use X variables to predict the Y variable, okay? Obviously, not every one of them is going to be very highly predictive. We're going to find the most highly predictive. And there are two that turned out to be quite good. One is called LoRa. This is an abbreviation for loss of rim area, so LORM, right? Loss of rim area measured by fundus photography. There are no missing values. So I think it's like, if you have glaucoma, your angle of vision, where field of vision is narrower, right? So loss of rim area is a measurement of how much your area is reduced. Then there's CLV, stands for corrected loss variance. This is another variable, and there are 12 missing values, okay? So this is the tree that we're talking about, right? This is the model. So in these slides, you will see a lot of pictures like that, and let me just explain to you how to read this picture. At each node, first of all, the nodes are numbered 1, and then 2, 3. So if the node is 1, this left node is 2 times 1, and the right node is 2 times 1 plus 1. Okay? So if this is node 3, this is 2 times 3 is 6, and this is 6 plus 1. So if a node is numbered k, this is 2 times k, and this is 2 times k plus 1. Right? So I don't number the nodes consecutively. So you see a 10 and 11, you don't see a 9, you don't see a 8. Right? I number it in terms of this kind of address, so that if you look, if somebody say, I'm looking at node 11, what is the parent node on node 11? You do integer division by 2, and that's 5. Right? What is the parent of 5? 5 is integer divided by 2, that's 2. Okay? So this way, at least, I can track where I am, right? Rather than just number them sequentially. Okay? So at every node, this is called an intermediate node. These bottom nodes are called terminal nodes, right? At each node, there's a condition. Here is LoRa. LoRa is an X variable, one of the 64 variables. It says if it's less than or equal to 56.4, you go to the left, right? So the condition, if the condition is satisfied, you go to the left. If the condition is not satisfied, you go to the right. Okay, that's why I put the condition on the left. Okay, yes, the condition, you go to the left. So anybody with this variable, loss of rim area, less than or equal to 56.4, go to the left. And then this is the CLV value. And if CLV value is less than or equal to 8.4, or missing. Okay, so now you see this inequality has an asterisk. This inequality does not have an asterisk. Right? If it has an asterisk, it means missing values will go to the left. If it doesn't have an asterisk, it means missing values go to the right. Okay, so this one has no missing value, has no asterisk. So now, if somebody, you're supposed to use this model to predict whether somebody has glaucoma. So now you have a new patient, right? The new patient has all these values, right? And you find out, okay, what is the LORA, what is LoRa for this patient? If this patient's LoRa is less than 56.4, you go down to the left. If this patient's LoRa is bigger than 56.4, you go to the right. What happens if this patient doesn't have LoRa? Maybe the doctor never measured that, right? then the patient will go to the right because the asterisk is not there. The asterisk is where the missing is. So whereas if this patient has LoRa 56.4, comes here, and then the patient is missing CLV, then the star here means you go down there. All right? So there's no need to impute missing variables. Okay? And at the terminal node, they are colored. Normal is colored blue. So these people are normal people. These are also normal. And these are the glaucoma people. All right? So here it says addition space. So you can read this caption. Right? Basically says what I say. But then there are two numbers on the side. They are the sample sizes for the classical glaucoma and normal. So here there are 77 glaucoma patients and 5 normal patients. So we predict everybody to be glaucoma. So our error rate in this node is 5 out of 82. Okay? So we make an error rate of 5 out of 82. Over here, the glaucoma patients, there's 1 glaucoma patient and 14 normal patients. So we make one mistake here, error of 1. Right? Which one is the top and which is bottom? Well, the algorithm will alphabetize this, put this in alphabetical order. Glaucoma is before normal because G is before N. So the first number is the first one. Okay? So this tree shows that this node has no errors. There's 0. Everybody is normal. Here is 0. There's 4. And this is all glaucoma. So the total number of mistakes we make here is 6. All right? So with this model, we make 6 mistakes. I chose this example because of the 64 variables, there are only 2 that appear. Right? The algorithm actually looks for all the variables, and we found 2. And I choose this because I found 2 so that I can show you the picture. Right? So this is a tree diagram, and this is the actual partition, right, in the 2 variables that I've chosen. So the first split is LoRa 56.4. That's this vertical line. Right? LoRa is left-right. LoRa is horizontal axis. So 56.4 is this solid line. Right? So if you're above 56.4, you go down here. You go down here, your next split on CLV less than or equal to 2. So CLV is this variable, and CLV, this is 2. Right? This is 2. Okay? So this node here is all red, mostly all red. There are some blues here. And that's why it's 77 versus 5. There are 5 here, but these 5 are almost on the border. Okay? So this rectangle corresponds to this node. This rectangle at the bottom corresponds to that node. So there are 15 points in here, and one of them, you see the red here? Right? You see the red here? Right? That's that one guy here. Right? That's one person here. Okay? So on the other side is when LoRa is less than or equal to 56.4, this half space. The next guy is CLV at 8.4. So CLV at 8.4 is here. Right? If you're less than that, then you're in here, and they're all blue. Right? They're all blue. 62 points, all blue. Okay? Above 8.4, you cut one more time at LoRa, so you cut here. All right? So there are 7 red people here, red points here. There's 7, and then the rest here are just 4. Okay? So this is called recursive partitioning. So one of the traditional names for this method is recursive partitioning. So you partition the space. All right? But you may think that, oh, this is easy. Yeah, maybe easy if there's 2-dimensional space. Right? But we have 64-dimensional space. So you need some machine learning software to partition in 64-dimensional space to find out what is the best first partition. So the best first partition is on this variable. Okay? All right? There's another thing here that if I tell you this picture is not truthful, would you believe me? You see, you look at this thing, you say, oh, yeah, I can draw this like that. Right? So they're equivalent. Right? They're not equivalent because there are 17, there are 12 values missing from CLV and no missing from LoRa. So this picture at the bottom is missing 12 points. In other words, they're not equivalent. Okay? The tree structure is more complete. It uses 100% of data. This plot is missing 12 points. Because I don't know how to plot those 12 points, because those 12 people are missing CLV. CLV is the y-axis. I know the x-axis because everybody has a LoRa. But there are 12 points without a y-axis. So where do you put it? You can't put it. Right? And luckily, you only have 12. If you have more, you'll see fewer and fewer points, and you find that they don't tally. If you add up all the points, they don't add up here. Right? So graphics is, people like to look at graphs, but graphs sometimes don't tell you the truth. And we may not even know if they're missing data. You use R, you plot. R will just plot whatever is not missing. And people, make judgments based on those plots. Okay? So that's why I say missing values is very difficult. It's very difficult. Okay, now, if you look at this, some people may say, well, can I change the splits from vertical and horizontal if I can have an oblique split, right? Maybe I get a better predictor, right? Well, an oblique split in 64 dimensions is no joke, right? You find an oblique split in 64 dimensions. 2 dimensions, maybe you can try. So what Guide does is, Guide does not try to get an oblique split in 64 dimensions. Instead, Guide will find the best 2 dimensions to do a 2-dimensional linear split. And it turns out that the best 2 variables are, again, CLV and LoRa. And so this is an oblique split. And it actually does worse, right? It does worse because there are 14 errors on this side. There are 14 blues. There's 7 blues over here, 14 blues, and there are 2 reds. These are 2 reds here, right? So the total number of points that are misclassified is 16. Whereas over here, there's just 6, okay? Although you have to be careful if you just look at this. This is called the resubstitution estimate. This is the apparent error rate. You grow the tree and then you put the data back down the tree and see how many are mistakes, right? Of course, if you keep splitting until every node only has 1, then you have 0 error, okay? That's overfitting. Now, so Guide avoids overfitting by doing cross-validation to prune the tree. So later on, we'll see how it does. It grows a very big tree and then it cuts it back, okay? So before we see this tree, we actually see a very big tree, but it cuts it back. And in here, also, initially, there's a very big tree, each time cutting on obliquely on 2 variables. But the pruning says that, well, it cannot sustain that error rate. It will just be overfitting, so it cuts it back. Okay? So for classification trees, no method can give you linear splits. If you want linear splits, you have to go with Guide. Okay, so that's one example. Okay, we're supposed to stop at 3:15, is that right? Our moderator has disappeared. Okay, so that is a tame problem. Right? It's not too bad. It's 12 missing values, not too bad. Continuous variables. Now let's look at another variable with all categorical variables. Okay. By the way, I should say in this example, you can think of logistic regression, right? Because it's predicting glaucoma or not glaucoma, so you can think it's 0, 1. So you can try logistic regression, but you only have 170 observations, 17 are missing, so you have only 153 observations, 64 dimensions. So what happens here when you do this is you get complete separation in 64-dimensional space, right? You see, logistic regression tries to fit an S-curve. You all know about that, right? So you have an X variable, and then you have some points where Y is 1 and some points where Y is 0. What happens when the 1s and the Ys are completely separated? How do you fit an S-curve? You cannot, right? It will not converge. The S-curve is going to go to infinity, right? This doesn't happen in 1 dimension. In 64 dimensions, almost certainly it's going to happen. Okay, so you cannot fit logistic regression. Forget about missing data. Even if you're willing to throw away the 17 observations, you cannot fit logistic regression, right? Okay, so let's look at this one. So we're going to also talk about logistic regression here. So we have 310 peptides now. This is by sample size n, right? In the glaucoma, we have 170 patients, so n was 170. Now my n is about double that, 310. And 181 of them will bind to a certain type of molecule, and 129 do not. So think of this as the equivalent of glaucoma patients and the normal patients, right? We're going to predict this type versus that type of peptides. All right? Peptides are short chains of amino acids. Okay? A class 1 molecule are cell surface proteins that present foreign peptides as targets. Right? So a class 1 molecule will sort of attract foreign peptides, so to eat them. Right? So now if you can bind them to some toxic drug, then these foreign particles eat them and get killed. That's the idea. One way to develop a drug. Right? So the idea here is, how do we know which peptide peptide would bind and which would not bind, right? So now, so what kind of data do we have? Each peptide is an amino acid sequence of length 8, all right? So so you imagine each peptide having 8 values, right? So x1 to x8. Okay, so now again, if you think of a data matrix, we have a y column. The y column is whether it binds or does not bind, right? So the y column, instead of glaucoma, normal, now is bind or not bind. Then we have, previously we have 64 columns of x, right? In the glaucoma example. Now we only have 8. Only 8. But these 8 are not continuous variables. Each of them is a categorical variable, taking values like A, B, C, D, and so forth, right? So we start with 8 dimensions, but each of these x variables can take up to 18 to 20 alphabetical values, right? So the question is, can you build a model that will predict whether the peptide is of this class or that class, right? So the data is actually in there, but you can download my data files and it's in there. So this gives you an idea of the data matrix, what it looks like, right? I cannot show you all 310 rows. I just show you the first 4 rows and last 4 rows. This is the ID number, which we won't care about. Then there's binder. Here I call them 1s and 0s, right? So it's a standard logistic regression. Now I have x1, x2, x3, x4, up to x8, right? I call them position 1, position. So each peptide sequence is a sequence of 8 characters, right? So for the first observation, the first position has a letter S, second position has a letter S, then position P, third position letter P, and so forth, right? The next one, again, S, and up to the fourth one, the first position is Y. So think of this as X1, X2, X3, and so forth, right? That's a data matrix, okay? So you think of this as sort of standard logistic regression, right? What do you do with logistic? What do you do in logistic regression when your X variables are categorical? You create dummy variables, indicator variables, right? But how many letters are there in each column? There are a total of 18 different letters in column 1. There are a total of 20 letters in column 2, and so forth. So you use logistic regression, you will have 17 dummy variables. X1 turns out 17 zero ones. X2 will turn out to be 19 zero ones. X3 will be 19 zero ones. So all of a sudden, from 8 dimensions, you have this huge number of dimensions. Okay? That is what classical logistic regression would require you to do. We have no missing data here, so at least that problem is not there. Okay? So if you use logistic regression, you end up with 149 indicator variables. Okay? We only have 310 observations, right? And sample size n is 310, but we have 149 dimensions. You start with 8 dimensions, suddenly we get 149 dimensions because you have to code the letters to zeros and ones, right? Well, in 149 dimensions with 310 points, you have separation. So you cannot fit a logistic regression model. Okay, even though you still have enough degrees of freedom to fit a logistic regression model, that model doesn't converge. Okay? And besides, if you think, when we talk about logistic regression, we're assuming there's no interaction term, right? I mean, in real data, why should there be no interactions? Well, you can't handle interactions for sure, because even without interactions, it doesn't converge. Okay? So you want to use lasso, right? Suppose you say, oh, we use lasso. This is large P, small n, use lasso. Okay? We use lasso, you get this result. So the idea here is which position is predictive of the status of the peptide, right? There are 8 positions. Which position is predictive? Use lasso, you see every position appears. Some indicators appear, right? Some indicators appear. So you want to say, is position 1 important? Well, position 1, level C, has a non-zero coefficient for lasso. Position 1, level indicator for D, has a coefficient that's not 0 for lasso. That's position 1. Position 2 is here. Position 3. So every position appears to be important. All right? Okay. How many of you have used lasso? Okay, no one has. You have used. How do you tune lasso? That's lambda, right? Use cross-validation, right? You use cross-validation, you run it again, you get different results. You get a randomized solution. Right? There's a lambda parameter in Lasso, penalty. People use cross-validation. Cross-validation depends on a random seed. You run it once, next second you run it again, same data, different results. Okay? The predictions may be okay. If you're trying to predict Y hat, maybe okay. But you try to interpret the model, forget it. You cannot even replicate in front of FDA. The FDA itself cannot even replicate. Right? But there's a lot of theory about Lasso. You look at Annals of Statistics, a lot of people write theories about Lasso. Okay? They ignore this cross-validation. Okay? So anyway, even if you, of course, one way to get around the cross-validation is do n-fold cross-validation, right? Then there's no randomness. But I'm just saying this for people who use Lasso routinely. Be careful, okay? Because people really know can challenge you. And then you look foolish that you didn't realize this. Okay? So use n-fold cross-validation, leave one out cross-validation, okay, there's no randomness. Right? But nonetheless, even if you do that, it doesn't tell you anything about what position is predictive of the status of binding, right? Well, this example became famous because in 1998, a group of computer scientists tried to demonstrate neural networks on this. Okay, this 1998 was a long time ago. To use neural networks, you cannot handle categorical variables, right? You cannot. So you have they call this, computer scientists say encoding, which is basically turning them into dummy variables. You have to encode them into numerical variables. And then the neural network requires tuning. Right? It's worse than cross-validation because it's too expensive maybe. So they randomly split the data into two sets, and then they build a neural network model. A neural network model has a lot of parameters you have to decide on. Right? Because a neural network is actually a multi-layer logistic regression model. Okay, so you have to decide on how many inputs and how many outputs and so forth. So the way to do this is they use this set-aside tuning set to see how many levels they need, to optimize the predictions. Okay, right? So immediately if you use neural networks, you're already working with a sample size smaller than 310 because it will save aside some for tuning. Okay, so not using a full sample size. Encoding into numerical variables, forget it, right? Because logistic regression doesn't converge. A neural network is a multi-layer logistic regression model with a smaller sample size. Of course, it will not converge, right? So what did they do then? Of course, then they converted them into some. So this is the same thing, logistic regression. This really doesn't work, all right? Then they brought in some additional knowledge from the literature that's not in the data, okay? The chemistry literature about these peptides. And they managed to classify each acid into 10 hierarchical clusters and then code each acid with. So they had two other methods. This one uses some physical chemical properties of the peptides like volume, bulkiness, flexibility, and so forth. So to solve this problem, they added some additional information that is not in the data. Okay? So for example, this is the second method, right? So they replaced the alphabets A, B, C, D into 80, 0, 1 variables. So for example, any position that has a letter A is considered to be in the hydrophobic cluster, right? So anytime a alphabet, so like look at all these alphabets, right? So all these alphabets, where I don't have any A here. So suppose look at S, right? S appears everywhere. So every time if S appears here in position 1, then S is in here, is in cluster 3, is also in cluster 6, is also in cluster 7. They're hierarchical clusters, right? So they sort of, they use some information to replace the categorical variables into these kind of variables. Then there's scheme 3, which is replace each acid by 13 chemical and physical properties, and they end up with 104 variables in total. So these are the, I don't have all of them, but so if it's A, then the volume has this value. If it's C, this volume has this value, and so forth. So they replace the positions with a volume variable, bulkiness, and so forth, right? So this is very smart stuff, okay? But you need this information. If you just use the data, you can't do what it do. So let's just look at the data now. Right? Let's go back and look at the data. These are just bar plots, distributions of the data. So the binders I paint in red, the non-binders I paint in yellow, right? So this is the distribution of binders and non-binders for position 1. Okay? So in case so this is position 1, right? There are a lot of S's here, right? You see a lot of S's here, right? So you look at position 1, right? The S is very long, right? You see that? S is very long because many of them have S. Even here, you see many of them have S, right? Okay, so that the S bar is very long, but some of the S's belong to y equals 0, y equals 1. Some of the S's belong to y equals 0, right? So you see that many of them are reds. That's a binder, and there are some yellows. Okay, right? And position 2 is like that. Position 3 and position 4. Okay, so now let me ask you, of these 4 positions, which is most predictive of the color yellow or red? So now I give you this data. This is the complete data, right, in graphical form. And now I come in with a new peptide. Okay, I say if you can predict whether this is red or yellow, okay, this is a new one, right? I'll give you $1 million. Okay, if you can predict whether this is red or yellow, I'll give you $1 million. But you can only ask one question. What question will you ask? The question you will ask is the value of position 1, 2, 3, or 4. Okay, you're allowed to ask either position. You can ask me, what is the value of position 1 in the peptide you're holding, right? Or you can ask me, what is the value of position 2 in the peptide you're holding, right? So which one will you ask? Position 1, 2, or 3? Position 1, right? Why? Why? Huh? Sorry? Yeah, why? Because they're mostly red. Yeah. What is the worst position? It's not here. The worst one is every bar is 50% red, 50% yellow. Agreed? Right? If I have a position, such that every bar is 50% red, 50% yellow, you ask me, you have 50% chance of being right, 50% chance wrong. The best one is some bars are solid red, some bars are solid yellow, right? If that happens, you are a millionaire. Right? Right? I don't have that here, that's why we're not millionaires, right? But what I'm saying is, if there's one of these positions where each bar is either solid red, doesn't matter whether long or short, right? Each bar is solid red, and the other bars are solid yellow. Then it's easy to predict, right? You ask me, what is the letter in that position? I say, is that one? And if that one is solid red, well, predict red, right? Okay, so this is only 4. There are 8. I cannot show all 8, so I'll show you the next 4. Now, which one is the best? 5? Anybody else has another? 8? Right? 5 and 8 are pretty close, right? 5 and 8 are pretty close. Right? So the machine learning algorithm that I'm going to tell you about in detail later is nothing but, can be boiled down to this, right? Patterns. Okay? And you have just proven to yourself that you're really good. Your eye and brain really good, right? You can guess, earlier you can guess position 1. Even when I ask you why, only one person could tell me why. But all of you guess right. You don't even understand why you guess it right. It's automatic. You see it, your brain tells you, oh, that one. But when I ask you why, you can't answer, right? Isn't that marvelous? Okay, so I tried to replicate this eye-brain thing into a computer program, right? The trouble is the computer has no eyes. So the computer cannot see these pictures, right? So how do you train a computer to come with the same answer that you got. When you don't show a picture to the computer, even if you show the computer a picture, no use, right? You take a selfie, no, the computer is not seeing anything, right? The computer has a camera, but it's not seeing anything. It's just taking a picture. Okay? So how do we write an algorithm that will at least closely reproduce this? So, okay, so the simplest thing, okay, so here's a secret when you do machine learning. A machine learning algorithm tends to be very computational, right? You heard of deep learning. Have you heard of deep learning? There's a very deep kind of neural network. So deep learners take a lot of time. Recursive partitioning can take a lot of time. At each step, you don't want to spend too much time. Because you spend too much time, the whole algorithm will take a long time to run, right? So our first step is to tell the algorithm that choose a variable, position 1, 2, 3, 4, 5, 6, 7, 8, one of them, and split the tree. Just like the glaucoma example, just like the peptide example, just like the glaucoma example. And after you choose that variable, then you decide what letters go to left, what letters go to right. Okay? So I'll show you the answer first. That's the answer. All right? So this is what guide gives. This is what guide gives when you have all this additional clustering information. Right? For now, let's deal with this. Right? Guide says, if position 5 is F, M, or Y, then you go to the left, and 159 of them are reds, and 10 of them are yellow. So majority is red, I paint it red. On the right side, 119 are yellow, and 22 are yellow. So this is yellow, and this is red, so I paint it yellow. Right? So this one makes 32 errors. Okay? And so don't be surprised if you don't get 0 error. If you get 0 error, you're going to overfit. Right? We only have 310 observations, right? So you should expect some errors. How can you be perfect? Right? So the guide solution says that this is the best you can do. If you split some more, your error rate, or true error will go up. F, M, and Y, right? So let's go back to position 5, F, M, and Y. F, where is F? F is a very long line, right? That's F. M is this short line, but it's still majority red, right? Can you see that? And Y is a super long line, majority red. So these 3 letters are super red. Not 100% red, but super red. Oh, the others have less red. So that's how the guide solution found it, right? But the algorithm has to consider between position 1, which we thought pretty good, right? And position 5 and 8, right? So here already some of you are fighting with each other about which one is better, right? How about you compare position 5 with position 1? So you see, the brain and eye are very good to do crude things. But when you have ties, the brain and eye will have a problem. Because you don't have an objective criterion. What you need is a numerical criterion, split ties. You just look, right? You know, you go outside, look, oh, everybody looks really nice. Or everybody looks really handsome or really pretty. But if you ask you to rank them, then you have a hard time. Like movies, right? You ask you like this movie? Yeah. You like this movie? Yeah. Rank them? Oh my God, how to rank them? They're all good. But a machine learning algorithm has to rank them. Has to choose the best, right? So how are we able to rank them? So we want to rank them in order so that the first rank is the one which has more bars, red and yellow. Second one, most bars, red. Oh, I'm out of time. Okay. All right. So maybe, okay, I'll just tell you how it does it. Okay. Very simple. Chi-square test. The chi-square test, right? You take position 1. Position 1, so we have 310 points. I draw a chi-square table. 2 rows and whatever number of columns, right? I do a chi-square test. And my chi-square is 185.25, degrees of freedom P17, p-value is less than 2.2 to the minus 16, according to R. Position 2, chi-square test again, right? Now the p-value is a bit bigger. Position 3, this is minus 15. This is even worse, right? Position 4 is even worse. So if position 4 is half yellow, half red, then the p-value will be very large, right? So that's all that guide does. Chi-square test. Everybody knows about chi-square test. You are looking at a correlation between color and alphabet. Right? I mean, if you sit down and think about it, that's what it's all about, right? That's how you could pick it out, because some bars are solid red, and the more bars are solid red and other bars solid yellow. That's most highly correlated with color, right? So we just use the chi-square test. Okay? So yeah, I think we should have a break. So we come back in, is it 15 minutes? Did we have 15 minutes or half an hour? Winky, do you remember? 30 minutes? Okay, 30 minutes. So, well, come back 3:45. Okay? Don't go to Chinatown, come back.